This video covers false prediction number 1. Now, as for the order of these false predictions of evolution, I'm simply following the order in the paper. It's linked down below. You can see that. So false prediction number 1 is the DNA code is not special. In this video, I will explain why this was a prediction of evolution, how and why it was falsified, and then what the result was, how evolutionists are responding to this false prediction. So let's have a look. First, what is the DNA code? It will help to understand a little bit about the code to appreciate this false prediction. You'll often see diagrams like this depicting the code, but what exactly is going on? So here is an illustration of what is going on when the DNA code is used to synthesize proteins in the cell. First, you have genetic material. This is a copy of a gene being pushed through a machine called the ribosome. tRNA molecules line up to deliver amino acids. So the input is the copy of the gene, which is a sequence of nucleotides, abbreviated A, T, G, and C. And then the output is a string of amino acids attached to each other, a linear string that will form a protein. So you literally are translating between a sequence of nucleotides to a sequence of amino acids. Let's home in here on just an example and see exactly how this works. So look at that tRNA there. It's carrying lysine as its amino acid. And you'll notice that it has these 3 nucleotides sticking out the bottom, and they mate up with 3 nucleotides in the copy, the gene copy. So the gene copy in that instance is AAA for adenine, 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 and they mate up with uracil, uracil, uracil. So the A is mating up with the U's. And the T, though, so whenever you have an AAA, it will attract a tRNA with 3 uracils sticking out the bottom, and that tRNA will always be carrying a lysine amino acid. Now, I say always, there can be errors and variations, but nominally, if things are going as they usually go, if you specify 3 A's in a gene, you're going to end up with a lysine. So that's how the DNA code works. Let's have a look here at the table there in the lower right. You see the AAA triplet of nucleotides corresponding to lysine. So that's what the DNA code, that's what that table is indicating, how those amino acids correspond to the nucleotide codes in the genes. And so there's your DNA code, all 64 triplets and the 20 different amino acids that match up to them. That's the DNA code. Now, you might be wondering, how do the tRNA molecules get their amino acids in the first place? How did the lysine get attached to that tRNA? How do the other amino acids get attached to the other tRNAs, and how is that done correctly such that you always have the right amino acid on that tRNA? Well, special proteins charge the tRNA molecules with their cognate amino acid. So there is a small army of proteins that go around and charge those tRNA molecules. Let's go back to that diagram. You'll notice right there in the upper left, there is the tRNA that's coming off that is spent. It's uncharged. It's lost its amino acid. So it will be recharged by one of those special proteins, and it's got to be the right amino acid. In this case, tryptophan is going to be attached to that tRNA molecule. Okay, back to where we were. So special proteins charge the tRNA molecules with their cognate amino acids. That's the magic. Those proteins are what enact the DNA code. Those special proteins are what make it happen. So proteins are required in this whole process. Those special proteins that charge the tRNA molecules are one example. There are many other steps in this process. The ribosome, you saw the ribosome machine right there, has a lot of proteins in it. So there's several steps in this whole process of making protein synthesis that requires proteins. So proteins are required to make proteins? Yes, proteins are required to make proteins. Yes, the DNA code is used to make proteins, but the code requires proteins. So you have a chicken and the egg problem. Doesn't work very well for an evolutionary narrative. This is not the sort of thing that evolution explains well. But let's set that aside. That's not what we're talking about in this video. Let's get back to our prediction. Now that we have an idea of how the DNA code works, its level of complexity and so forth, what about this prediction that we're talking about? What's going on? So why was the DNA code predicted to be not special? That's the prediction we're talking about. The DNA code is not special. Why did evolutionists say that in the first place? Okay, so back to our code here. Now, this is the evolutionary tree, a notional look at an evolutionary tree. You see time is going up there. The blue dots represent today's species, today's extant species, the ones we see alive today. Down at the bottom, you have the universal common ancestor of all life, the last universal common ancestor, Luca, of all life. Now, the way evolution works is if you have some design that's common between neighboring species, as those yellow circles suggest, then according to evolution, that common design must have been inherited from the common ancestor. If you have a common design more broadly present in more species that are neighboring each other, then you have to go back farther in time to the common ancestor. That's the way evolutionary theory works. Designs are inherited from the common ancestor. That's the whole idea. If that's not working, then evolution isn't working. Now, what if you have a common design across all the species, as all these yellow circles suggest? Well, that's the case with the DNA code. The DNA code, interestingly, is found in all species, with some minor exceptions here and there. But the standard code is overwhelmingly found in all species. You look at the mammals, the reptiles, the birds, the fish, single-celled, unicellular species, they all share this same standard genetic code. So what does that say? Well, according to evolutionary theory, it must have been present in the common ancestor of all life, the Luca, last universal common ancestor. So there you have it. It evolved from a common ancestor. The DNA code must have been present very early in evolutionary history. And then, interestingly, after that, it must not have changed. So over billions of years where you start with this crude initial single cell rudimentary organism, and then creating everything we see, the tens of millions of species, all the different designs, all the different mechanisms, and yet the DNA code does not change. So that tells, according to evolution, that tells them that the DNA code must not be evolvable, which kind of makes sense. If you have a code that's coding for thousands, tens of thousands of proteins in an organism, if you were to go change the code, that's going to wreak havoc amongst all these different genes that have already been set up. So it's like any code, you change the code and that's going to screw up the messages. So, okay, it's not evolvable. So those are the two conclusions there. Evolution must conclude, one, the genetic code arose very early in evolutionary history, back when things were crude and rudimentary and so forth. And the genetic code is not evolvable. So what does that add up to? Well, when I say it's not evolvable, by the way, that takes natural selection out of the picture. So you're not, usually the narrative, the explanatory mechanism evolutionists use for highly complex, sophisticated, fantastic designs is, oh, natural selection did that. Oh, isn't that cool what natural selection did? But that's natural selection for you. It does everything. So no matter how optimal, how fantastic a design is, natural selection did that. Well, that's taken out of the picture here. You no longer have natural selection as an explanon, and so that forces evolutionists to conclude the DNA code just is something that just somehow arose without being optimized or going through any selection process. It was early in the process of evolution, and it was not optimized. So it must not be special. It must be mundane. It's just one of all the possible codes. It's one of those codes. And you can imagine, I didn't mention this, but going back to that table that I showed you, there's a lot of different codes that are possible, theoretically at least. If you just were to rearrange the table, you could rearrange that in an astronomical number of ways. Think of all the ways you could rearrange that table. We just happen to have that one. So the code must not be special was a prevalent, common, well-accepted prediction of evolutionary theory in the, say, 1970s, 80s, 90s, so forth. Let me give you a few examples of this. Let's start with Francis Crick, who described the DNA code as a frozen accident. It arose from a series of happy accidents. It was just a frozen accident. Frozen meaning, it's not evolvable. Accident meaning it's just one of many codes. It's a mundane code. It's not optimized. Francis Crick, Nobel laureate, co-founder of the DNA structure. Here's a very popular undergraduate textbook on molecular biology. Quote, "The code seems to have been selected arbitrarily, subject to some constraints, perhaps." So again, this idea that it's arbitrary, it's not optimized or even refined, it's just arbitrary. Another textbook, an evolution textbook, "The code is then what Crick called a frozen accident. The original choice of a code was an accident, but once it had evolved, it would be strongly maintained." So it's kind of unusual wording there too. Note the wording choice, the original choice. Well, of course, it wasn't a choice. Evolution is not a conscious agent. But those are some examples of how evolution looks at the code as just not special. So how was the prediction falsified? Well, in 1999, this paper came out. The genetic code is 1 in a million. 1 in a million was a figure they arrived at by looking at its error correction capabilities, error correction features. A later paper refined that and came up with a much stronger number. Instead of 1 in a million, it was 1 in a million, and more. Quote, "We show that the probability of obtaining a genetic code with a cost as low as that of the standard genetic code, by chance, is 10 to the minus 20th, much lower than the previous estimate." So yes, it is a very special code, very, very, very special code. The code is not mundane. And even this paper did not look at every possible angle on the code. There could be more aspects of the code making it special. In fact, other papers have hinted at that and talked about that. Here's a paper that says that the DNA code is nearly optimal for encoding multiple layers of information. Here's another paper that says the code optimizes, quote, "a combination of several different functions simultaneously. We wonder what other remarkable properties it may bear." So obviously this prediction of evolution did not last, did not stand. It was falsified. So, okay, we've got a prediction of evolutionary theory, clearly stated, clearly held by leading authorities, and it was clearly falsified, no question about it. It is false. Now, what does that mean? What does that lead to? What did the false prediction lead to? Let's have a look. So, in the years since, a multiplicity of hypotheses have been suggested by evolutionists. Of course, there's Crick's frozen accident hypothesis. Then there was the stereochemical hypothesis, the coding enzyme handle hypothesis, the coevolution hypothesis, the four-column hypothesis, and the error minimization hypothesis. A lot of different hypotheses floating around. Well, all of these hypotheses are problematic. For example, error minimization. The error minimization hypothesis, well, first of all, requires that the code is evolvable. You are evolving the code to get better and better error minimization. So it's definitely a random mutation with natural selection sort of narrative, the typical, usual evolutionary narrative. But we just talked about earlier how that's such a problematic idea because it must be frozen. So right off the bat, you have a problem here that this hypothesis is requiring the code to be evolvable in the first place. But secondly, the code does not appear to be at an optimum point for error minimization. In other words, it's not at a local fitness peak. There are better codes. As good as the code looks, according to those papers I showed you, there also are better codes. So if the code arose by error minimization, why is it not better? Maybe evolution just got stopped in its tracks. Okay, maybe, but there is that question. Finally, the code displays a structure and patterns that are not related to error minimization. So it can't be just error minimization driving evolution to create the code. There's this other things going on in the code that have nothing to do with error minimization. So there's problems with the error minimization hypothesis all around, just like all the other hypotheses. So another idea is, we've got some hints at error minimization, but maybe there's also coevolution going on and stereochemical and frozen accident, and let's combine these as we need them, kind of a menu approach. Pick and choose from your favorite hypothesis when and if you need them. So a combination of these has been floated as an idea. So you have a multiplicity of hypotheses, and the reason you have a multiplicity is because there is no single good hypothesis, which is not a real good sign here. Evolution has really been struggling to explain the evolution of the DNA code. This false prediction did not lead to new knowledge or some sort of smoking gun that explained everything. Aha, this is it. Yes, the prediction was wrong, but that led us to now understand exactly how it works. Everything falls into place once we realize this. That's what you want to come out of a false prediction. We had to pay more attention to it, we focused on it, and aha, look what we found. That's not the case with evolutionary theory and, in this case, the DNA code. So here's some quotes from the literature to help illustrate this. Quote, "At the heart of this problem is a dreary, vicious circle. What would be the selective force behind the evolution of the extremely complex translation system before there were functional proteins?" Remember we talked about the chicken and the horse problem. Continuing, "And, of course, there could be no proteins without a sufficiently effective translation system. A variety of hypotheses have been proposed in attempts to break the circle, but so far none of these seems to be sufficiently coherent or enjoys sufficient support to claim the status of a real theory. Summarizing the state of the art in the study of the code evolution, we cannot escape considerable skepticism. It seems that the two-pronged fundamental question, why is the genetic code the way it is and how did it come to be, that was asked over 50 years ago at the dawn of molecular biology might remain pertinent even in another 50 years. In other words, there's no progress here. There's no fruitfulness. The false prediction doesn't reveal some new avenue, new pathway of thinking that leads to more fruitful discoveries. Let's look at another quote here, a little shorter. This one, "To be honest, we are stuck. Most papers on the origin of the genetic code are reviews, like this one, and not original research. It seems that the field has been stalled." So again, fruitfulness is a problem. So we have a failed prediction. We have a prediction that was made, we have a failure of the prediction, and we have the outcome being a lot of further problems, just kind of highlighting the failure, not resolving the failure. Okay, that's important. Failed predictions are important. But what are we going to do with it? So what I'm going to do in this paper and with these false predictions is use them as a framework, as fuel to look at failed theory virtues. And in particular, I'll look at these 8 theory virtues and see what the failed prediction portends for these failed theory virtues. And let's look at these theory virtues. Philosophers of science have come up with all kinds of virtues that a theory can fulfill. And here's these 8 I'll be using in this paper. Evidential accuracy, causal adequacy, internal consistency, internal coherence, universal coherence, simplicity, durability, and fruitfulness. So it's a good framework here that we can use to see what these failed predictions mean in particular, and not just making a long list of failed predictions, but what does it really mean here? And so let's look at these 8 virtues in more detail, just because I'll be using these throughout the paper and throughout this video series. Evidential accuracy. Well, a theory, T, fits the empirical evidence well. If that's the case, then it has evidential accuracy. The theory fits the empirical evidence well. Causal adequacy. T's causal factors plausibly produce the effects or the evidence in need of explanation. So in other words, the theory has certain explanations, the causal factors, forces, processes, what have you, that the theory calls upon. And those can plausibly produce the effects. Internal consistency. T's components are not contradictory. You don't have self-contradictory components in the theory. Internal coherence. T's components are coordinated into an intuitively plausible whole. T lacks ad hoc hypotheses. Theoretical components merely tacked on to solve isolated problems. Universal coherence. T sits well with or is not obviously contrary to other warranted beliefs. Simplicity. T explains the same facts as rivals but with less theoretical content. So it's simple. You're not just adding more and more and more theoretical components. Then you're violating Occam's razor. Durability. T has survived testing by successful prediction or plausible accommodation of new data. And then finally, fruitfulness. T has generated additional discovery by means such as successful novel prediction, unification, and non-ad hoc theoretical elaboration. So yes, you can elaborate, but it shouldn't be ad hoc. Well, as I read through that list of failed theory virtues, you may have been realizing, in this particular failed prediction, the DNA code is not special. The theory fails badly on almost every single one of these. I mean, the one that you might be able to excuse evolution on here would be the third one, internal consistency. Maybe that one isn't a problem in terms of this failed prediction, but all of the others are failing on this prediction. Evidential accuracy, the theory fits the empirical evidence well. Well, no, it isn't. Causal adequacy, it has causal factors to plausibly produce the effects. Well, no, that's the problem. It doesn't. And so on and so on. Simplicity, fruitfulness, durability, ad hoc explanation. It's failing on all of these. So I'll excuse the third one here, but this particular failed prediction brings to light failures on all of the others. It fails on evidential accuracy, causal adequacy, internal coherence, universal coherence, simplicity, durability, and fruitfulness. Religion drives science, and it matters.